Welcome, everybody. Welcome. I think we're kind of like straggling in a little bit. Good to see everybody. It's not officially summer, but it's plenty hot out here today. Not loving the heat, but hey, but it's here. All right. Another minute or so. In case you don't know, I'm Terri Conneran, and I'm just happy to see people today. You guys, I mean, anyway, I gotta say, we really have a nice looking group of people just in general. So like, thank you all for just showing up and just to brighten my day. It's kind of nice, it's kind of nice. And just as a little reminder, this meeting is being recorded. So if you are in the witness protection program or have some other reason that you do not wish for us to see your adorable face, please turn off your screen. And we will do our very best to continue protecting your witness protection program situation or what have you. And I'm trying to, do I need to do anything about speakers or anything, or should I just get rolling? Okay. Kind of a casual group with y'all today, and I am so completely unprepared, and I apologize. I have Dr. Melissa Johnson. I mean, I took the weekend off, and usually I'm over here on the weekends working, and so I didn't do it this weekend. I said, I need a little family time, and so normally I'm all prepared, and I did not prepare myself today. I have Dr. Melissa Johnson with us, who is from Terri Conneran. And I should have all your fancy titles and degrees and stuff up here on the screen. Okay. But we're just going to go with she's a super nice person. She's an amazing doctor. And we just and she also knows what it's like to just really have life. And so I'm really excited to have her join us today. And be able to kind of go through some questions and answers about ASCO and what have you. And before I let her do a quick intro, if y'all would be so kind as to mute your screens so we don't get any background noises. We love to see your pictures, but it's hard to hear over them. So please mute your screens. So thank you for joining us today, Dr. Johnson. Would you do a quick intro on who you are, where you are, and whatever, and we'll go from there.
Sure. Hi, everybody. Thanks for joining today. Glad to meet you all virtually. I'm the director of the Lung Cancer Research Program for Terri Conneran. Terri Conneran is a physician-based network. I'm based in Nashville, but we have clinics, phase 1 clinics, as well as phase 2-3 clinics all over the country. And so my job as the director of the Lung Cancer Research Program is to help supply clinical trials to all of our physician practices. And we can talk more about that if it's of interest. I did my fellowship training at Sloan Kettering with the thoracic group there. And so I've been a lung cancer doctor now for 13 years. Just taking care of lung cancer patients. My day job in Nashville is as a physician in a drug development unit. It's one of the places in the country where first-in-human trials are done. Terri Conneran put 1,500 patients on phase 1 trials last year, and probably 2/3 of those were in Nashville. So we're busy, and it's what a great opportunity it is to be among the first in the world to use drugs. Those that are going to become FDA approved usually present themselves early as promising, and those that are not also tend to present themselves early. So it's a wonderful thing to be able to provide these sorts of opportunities to my patients. I moved here 8 years ago. I moved my family to Nashville so I could take this position as the associate director of the Lung Cancer Research Program when I joined. But I moved from Northwestern in Chicago, where I was on the faculty. So I moved my husband and my daughter here. My husband had to change careers for the move, and we just over and over again, it's been a wonderful thing for us. So that's, and maybe I'll just say that I'm the only doctor in my family. I'm the daughter of a Marine Corps officer. My dad was in the Marine Corps for 35 years. My brother has been in the Marine Corps as long as I've been a physician. And I have cousins that were Marines. My grandfather served in World War II in Korea. My dad had a low draft number, and so he joined the Marine Corps as a second lieutenant, so he wouldn't get drafted into the Army as a private when he was in college. And so no one in my family knows medicine, and so I think I try to treat people the way I would treat somebody in my family. I try to talk to people as if they were in my family, so you can tell me to dial anything back, and I'll try again. Just because Terry and I were at ASCO last week, it's this big meeting. It was over 40,000 people were registered, which is pretty amazing since we haven't had one in person since 2019. And over 30,000 of them were there at the meeting. So when I come back from those meetings, like the science speak, the onc bot in me, as my husband will say, he's also not a doctor, is like tends to get a little out of control. So would be happy to talk about things that happened at the meeting or things that you're curious about otherwise.
Okay. Well, that's a good jumping off point, because I mean, we have we never really know who all is going to be in the meeting. And so we have all across, and you'd be surprised the onc geeks that we get here, okay, because we can stay really focused. But let me just start at the top, because I don't know what a physician-based network is, and I think that's just kind of interesting.
It is, because Sarah Cannon's network is to bring cutting-edge therapies to patients in the communities in which they live. So Sarah Cannon's network is all community-based. I had interviewed 3 times at Sarah Cannon before I realized I was going to a community practice. I had always thought that I would be an academic physician, meaning that I would do research. And that's what I found was the opportunity here to continue to bring cutting edge therapies to new patients. So the community-based practices that do research with Sarah Cannon as the research engine are Tennessee Oncology, of which I'm a member and a partner, Florida Cancer Specialists. So Tennessee Oncology has about 80 physicians across the state of Tennessee. Florida Cancer Specialists, about 200 medical oncologists across the state of Florida. And then we have about 3 or 4 AON practices, American Oncology Network practices, and they are in Columbus, Ohio, Asheville, North Carolina, Arkansas, Sangmeister, and I'm flummoxed because I'm forgetting one. I'll remember it. And then we have a couple academic partners. Sydney Kimmel is in Philadelphia, and Oklahoma University in Oklahoma City. And so the advantage, I would say, of a physician-based network began with Tony Greco and John Hainsworth, who were the founders of Sarah Cannon. And they actually left Vanderbilt in the early 90s went into practice in Tennessee Oncology, and just said, we want to get rid of all the bureaucracy of clinical research. And so for a very long time, Sarah Cannon was just about answering the questions that were important to the doctors taking care of patients. So often the trials that we select are really practical, common sense questions, the ones that for which we are wondering in our practice what we're doing. Over time, Skip Burris, who's my boss and the chief medical officer of Sarah Cannon, was recruited to come lead the drug development unit. And so our other distinction is as the first community practice to have phase 1 research, where there's a lot of safeguards that you have to build in to take good care of patients, but also to understand how the drug works. And to fast forward to 2022, we put 3,000 patients on clinical trials across both early and late phase last year alone. And that was right after COVID. So these are successful programs. Often our startup times are faster than the academic facilities. And for those of you who have been on a clinical trial, you know that sometimes you hear about a trial, you hear about a drug, but it takes 6 to 9 months to be able to participate because the paperwork takes so long. So that's another, I mean, and that's just a reality of doing research at any university, because there are more committees and more wickets that need to be crossed, I guess.
Okay.
So
okay.
that
so I guess an off point to kind of like even get into like all the like, you know, all the cool stuff that happened. And what you see is, you know, how it affects like any KRAS patient. Okay, from stuff that came out at ASCO. And if you want to start at the biggest thing first or the smallest thing first, I'm going to let you just lead the parade.
Well, maybe I'll say one other thing just about where I work, and that is that about 80% of patients are treated in the community. And so if we want to do something that impacts patients on a large scale, I believe that community research will be the way that we do that. A good example is the KRAS G12C inhibitor, for which we heard the results of the adagrasib trial. We participated in the trial. Alex Spira is from US Oncology, another community-based network, and he presented the work. The reason it's important is because so it's the second drug for patients with KRAS G12C mutations that is rapidly approaching the market. And you might say, well, I'm not a KRAS G12C, I'm a KRAS G12 something else. So I don't understand, why do they get 2 and I don't get any? I think the truth is that as we flush out how to target KRAS G12C effectively, the others will fall into line. And I can talk more about some of the research that we've heard about both at AACR this year and ASCO for those non-KRAS G12C cancers as we go. But the KRAS G12C inhibitor adagrasib showed response rates about 40%, PFS around 6 months, and overall survival between nine months and a year. And so it is in line with sotorasib that has achieved its FDA approval. A couple things that I think are really interesting about the two as we try to, they're probably tolerated about the same as well. I learned at this meeting that adagrasib's half-life is 24 hours. So that means it takes 24 hours for the drug to get out of a patient's system, it takes 24 hours for the level in a patient's body to go down by half. But the drug's dosed twice a day. Whereas sotorasib, half-life 6 hours, I think, and it's dosed once a day. So you have this adagrasib drug that you're getting every 12 hours, even though you really might not need it that often. And then the sotorasib half-life is shorter and you're getting a bigger dose, but only once a day. So to me, that's a big difference that nobody talked about. Because what we've learned is that, unfortunately, adagrasib and sotorasib don't work like osimertinib, for example, for EGFR positive patients, where the response rate is 60 to 80%. So where most patients are having a response, this drug works about 40% of the time with response rates. Patients have shrinkage 40% of the time. So that tells us that we're going to probably need to combine it with something. And we're going to need to combine it with lung cancer patients, just like colorectal cancer patients, pancreatic cancer patients, ovarian cancer patients. And that was something else that people didn't talk about very much, but I think is the next step. That was, Terri was nice enough to come to my talk early on Tuesday morning, the last day of ASCO. And she gave me some socks that I used for my team this morning as rewards for all of their hard work during that, like, you know, plugging and chugging, putting the data in. But so I think that's a big difference that we're not talking about. Now, the other thing is that adagrasib doesn't have an approval yet. And so there are rumors that it won't be approved at 600 BID, which is the dose that everyone, that the data was based on at ASCO, but rather at 400 BID, a little bit less.
BID is twice a day, right?
But sorry, yes, BID is twice a day. Okay. And we also hear that sotorasib is being even though it was approved at a dose of 960 once a day, it's being still evaluated at lower doses to see what the best dose is for that.
Because it wasn't that one of the things, we talked to a lot of patients that the dosage gets changed. It starts at one level and then gets changed back down. And I know there's different levels, and the FDA was kind of like, they were monitoring it. I don't remember what their terminology was, but something like, hey,
dose optimization.
There you go.
Is the
right?
term.
Yeah, they're monitoring it to, you know, so to see what's best for it. So do you think there's a possibility that they're going to be like changing all that around as far as like what the standard prescription is going to be?
I think it's, I believe that it's a step forward for sure, because it represents to me an acknowledgement that the best dose in terms of, you know, the numerical outcomes that you see in the trial may not be the best dose for each individual patient. There are other drugs. Afatinib is one that comes to mind. It's an older drug for EGFR positive patients. Its approved dose was 40 milligrams once a day. And I started everybody on 20. I started everyone on half the tolerated dose and then went up to 30 for patients that felt okay there. And I rarely went higher than 30. Sometimes I stayed at 20. So I think as a patient advocacy organization, this is a really interesting topic to watch, because as we talked about, I think we got into it a little bit during our session, Terri, at the end, that patients are afraid to really tell the doc how they're doing. Patients are afraid to acknowledge that they, you know, that whether they're able to take it every day. You know, there are situations like what Janet puts in the chat, where you know, the sotorasib is being lowered because of liver enzyme elevation, also renal insufficiency, and that can happen with adagrasib too. I don't want you to hear that it happens with one and not the other, but it means that dose is, there's an incredible amount of art to defining the dose, not just science. And that's actually the deceptive, one of the things that is deceptively interesting to me about being a phase 1 investigator. Because if you think about it, for every study after that, the work you do early on makes such a difference. And I think we're hearing from the FDA a recognition that we need to do better there.
Well, and I mean, every person's not the same. I mean, that's just the truth, right. You know, and what somebody at 100 pounds needs versus somebody 300 pounds, I mean, just mathematically doesn't make sense to me. So
even two people at 100 pounds, their ability to metabolize a drug can be very different. And, you know, those of you that need pain medicine, your ability to tolerate pain medicine can be very different from one to the other. And I say to all my patients, whether they're getting a clinical trial drug or if they're getting chemotherapy, I don't know how you'll do until we give this first dose. And so the hardest part is in reacting to what happens to you in this first cycle, and then we'll be able to move forward, you and I together. But for every patient, it's different.
Now, there was some interesting data that came out from adagrasib on brain mets. Could you kind of like go through that with us?
Yeah. Sure. So the Mirati is the company that makes adagrasib, and they had patients with treated brain metastasis that were included in the first in human study. And then they took a cohort of patients in that study who were brand new diagnosed patients who had not had treatment for existing brain metastasis. And they allowed those patients to join before they had their brains treated with radiation. So they had two groups. They were less than 30 patients each. One group that was within the full study who had brain metastasis that had radiation to those spots before they went on the study. And then they had another group of patients who hadn't had treatment. So the reason to look at those two in parallel was that they were able to show that the patients with untreated brain metastasis did very well with just the pill, suggesting that the pill was crossing the blood-brain barrier and controlling the cancer that was there. In fact, they had a couple different case studies that they reported of patients whose the amount of cancer that was in the brain certainly regressed without any radiation. So the reason that's, and the response rates in the brain were about 30%. So remember, I told you the response rates were about 40% in the body. So it's hard to know with just 30 patients, is that 30% of the time you're okay in the brain? Is that most of the time you're okay in the brain? Just that little 10% change is all you have to worry about? You know, there's more work to do, but it does tell me that if I had a patient who's new to me, who didn't want radiation and had limited cancer spots in the brain, I think I would feel comfortable trying a month of adagrasib and then rechecking the brain to see if we might be able to avoid radiation. It's not always possible, and sometimes radiation really is the way to stop cancer in the brain from spreading. So I think that it's it will probably need to be a nuanced decision, you know, an individual decision that a patient and the doctor make together. But it does give us confidence that it's okay to do.
Okay, so, and when you say a nuanced decision based on the data, but it's not approved for that, is that what's considered off-label?
Well, it's you're right. Adagrasib is not approved at all right now, okay. And we'll see whether it'll include whether it'll. I think it will not be approved based on 30 patients that were in this cohort. So when I say it's a nuanced decision, if a patient, it's a decision that a patient and his or her oncologist should make together. Because if you have 20 tiny little brain mets at the time of diagnosis, but you're asymptomatic, maybe you do a month of therapy and then recheck.
Yeah, and what I hear is 20 little tiny brain mets. There's no such thing as a tiny little brain met if it's my brain. Well, and that's the scary part.
So that's the nuance part, Terri, because as you talk to your oncologist, you would say, well, wait a minute, let's be careful here. But the way to treat 20 little cancer spots in the brain is whole brain radiation. So 10 fractions of treatment to the whole brain, not just the little spots. That for some patients can also have a risk of short-term memory decline. And so there will be patients that say, well, I'll wait a month. You're gonna scan me in again, and if it doesn't show improvement, then I do radiation. Okay. So, but that's, you know, that's a gut thing. So do you see what I mean? Mm-hmm. It's a nuanced decision.
Okay. And a lot of times, I mean, things just as soon as it hits the brain, a lot of times they're excluded from a clinical trial. And so this is kind of unusual that it was already included, isn't it?
most clinical trials will allow these days, early trials will allow patients with brain mets as long as they've been treated. And then there's a washout period typically. And this is where the washout period is very much in the, is very much to homogenize the clinical trial population, but not necessarily because it's the best thing for patients. Because if you need radiation to your whole brain, and I think the washout period was a month. So you get radiation and that takes 2 weeks. And then you have to wait a month until you can go on the trial. That's 6 weeks potentially without any therapy. Early on in somebody's disease course, that's a reason that I would say, okay, we can't wait. We can't wait that long for a trial. So we'll have to do a standard of care approach, and then we'll do a trial in the future. But the washout is the issue.
Okay. Okay. And now when you were talking about in the brain, are you talking about in the brain, not like on the skull, correct?
Correct.
Okay.
And
is the skull considered a bone met? Is that how it is? Or is
yes. Yeah, that's right. That's right. It can be. It can be a subtle brain met, but that usually takes special scans to figure out.
Okay. Okay. Now, all right. Now you were saying earlier that if we're not G12C, like me, I'm not G12C. Okay. How does all this stuff help me?
Well, a couple ways. Turns out there's more than one way to inhibit KRAS G12C. This first way was very much dependent on the configuration of the mutant KRAS G12C protein. And as the protein shifted back and forth, it exposed a vulnerability that allowed the drug to bind. But it turns out that there are other ways now we're learning to bind KRAS that bind KRAS irrespective of the type. There are these cool new drugs called GTP-on inhibitors. It's a tri-complex conformation, and it uses it basically catches the KRAS like this with the drug and a protein that's around a lot, ubiquitous, we say, and it stops the KRAS from cycling. And those drugs are being developed by Revolution Medicine. And there is one that is for KRAS G12C patients, and another that's for other KRAS mutant lung cancers.
Yeah, so there are also now those are the pathway drugs, correct?
Pathway drugs.
I mean, those are like the SOS1 and
no, they actually they're active KRAS inhibitors. So they actually bind to KRAS itself.
Okay.
There is a KRAS G12D drug that is being developed. So KRAS G12D is the most common KRAS mutation, especially in pancreatic and colorectal cancer as well as lung. There are protein degraders. So they identify the KRAS protein and bind and get rid of it. They're not in the clinic yet, but I am interested in those as well. And then there are drugs that hit downstream effectors in the same pathway as KRAS. So they don't affect KRAS, but like Eric is sharing, they inhibit ERK, which is two proteins downstream of KRAS. And they can also inhibit MEK, which is upstream of ERK, downstream of RAS. And there are still others as well. So there's a lot to be hopeful for. You shouldn't feel like you don't have you don't have as many options if you have a KRAS G12V, for example. We found that in general, the doctors, now that they're identifying patients that have a KRAS G12C, are also at least identifying the KRAS G12D or V or F, somebody put in the chat as well. And and are referring because of that. So I would say that that there's a lot to be hopeful for across the board. This was just the first, the what's the expression about the tip of the iceberg.
How long do you think it's going to take to get these next, besides the G12C, like the G12Ds and what have you, into the clinic or into clinical trial? I keep hearing almost.
We have our first KRAS G12D drug SIV, which is the opening of the trial this week. So they're there. And I would say Mirati, that made adagrasib, they also have a KRAS G12D that's not quite in the clinic, but they think it'll be in the clinic by the end of the year. So 6 months.
Now, which trial are you opening up this week, you said?
Astellas has a KRAS G12D inhibitor.
Okay, that was one of the trials that we had posted on in our group that is going to be coming soon, but we didn't know when it was going to be and where it's going to be. The last we were heard last week is, and like I said, I took the weekend off. So the last I saw last week, I didn't know where they were going to be, or at least I couldn't stop looking, one of the two.
Let me see if I could get the NCT number.
Yeah, we had that posted. And so I think
you do. Okay.
Yeah. Thank you though. Thank you. Okay. Yeah. Okay. All right. Does anybody have any questions that are like just burning? Okay. I see burning. It's like burning and you just can't wait. I have a question for Dr. Johnson about clinical trials in general. When a clinical trial is conducted at specific hospital locations, are there ever situations where a patient could visit their own local hospital where the trial may not be available to get blood work, scans, et cetera, done for the trial, and the coordinator doctor could participate via remote?
Yeah, that's something that we've been talking about. I think what exists today is, for example, for the adagrasib trial, it required weekly blood work. And the week, like forever, which when we look at a trial and consider it, the first thing that we'll say to a company is, this is not patient friendly. You cannot expect patients to come in weekly for blood work forever. And so they thought about it, and Mirati came back and changed, I think after 6 months, they took away one of the weekly blood draws every three weeks. So it was not very helpful. But what we were able to do during COVID, especially, is when patients traveled to Tennessee to be part of our trial, typically from regional states, Southern Indiana, Illinois, Mississippi, Alabama, Arkansas, certainly Kentucky. We put their local lab on our 1572, which is the paperwork that we need to do, so that we could do those lab work visits. They could do those locally. And then we did do telemedicine visits, obviously much more. Since COVID has finished and we're no longer able to do telemedicine outside of the state of Tennessee, that's changed that a little bit. But I can't believe that genie's going back in the bottle. I just think it's too, so I think, and all of that to say, we have increasing technologies that help us do this. So do we have the wherewithal, the desire to do that? I think patients certainly have that desire. And I think doctors certainly have that desire. If I could work with a doctor wherever and remote in, and he be seeing you in the clinic, I would absolutely do that. And in the past, what has gotten in the way is the pharmaceutical company that, doesn't want to take on that risk. Now at ASCO this year and also AACR, and in between, we've heard loud and clear from the FDA that late phase clinical trials need to be happening in 20% of our patient population, not just the 3% that go on trials. And so the onus is on all of us, but maybe especially the pharmaceutical companies that are funding these studies to figure out how they're going to do that. And so I think there's never been a time where we were closer to being able to do that Because
so oftentimes, like me or somebody else, a patient advocate will have the opportunity to review a clinical trial or something that's going to be patient facing, whether it's at phase 1 or whatever, and through the process. And that would be a valid thing for us to push back on. Is what you're saying. It would be totally valid for us to push back and say, well, why can't we do blood draws at my local, so I could just go to my corner, okay, as opposed to going even uptown. Just 30-minute drive compared to a 10-minute drive is a big time saver. So that would be the other thing for us to say.
I think that it starts with grassroots like this, like think globally, act locally is the way that will change this. I see from Eric in the chat, he still has to go to Boston every couple of weeks. It's still to travel 2 hours to Boston to get blood work done and to get more pills. In the current collaborations and research organizations that we have, that's what they've asked us to do. So it'll take some pushing, I think, but I think it's doable. And I think it's that seems to me to be low-hanging fruit about how we would improve clinical trial enrollment, because so much of it for our patients in Nashville, which is a pretty central place in the Southeast, is that doctor, I can't come all this way, even from Knoxville on the other side of the state, they don't want to come, because it's expensive, and it's expensive, not just in money, but in time.
Totally. And time is the most precious part that we really have as any sort of a cancer patient at all. I know OCE through the FDA, which is a lot of letters. I think OCE stands for like Oncology Center for Excellence or something. And they were saying that that's one of the things that we patients need to be pushing back on. And we need to be able to be vocal, I guess is what I'm trying to say. We're trying to be vocal about how important these things are for us and to us.
Yeah.
And so what I'm saying to the community and to you as well, that if there's opportunities for us to share that or work as a collaboration, we need to have those opportunities. And so let us know about them. And I'm sure that there's all sorts of red tape and all sorts of nuanced things that go on behind the scenes that just make things a lot more complicated, but we need to be able to do that on both fronts. So it's just kind of like throwing that out there. And patients that are in here and caregivers and people that just care about this need to be willing to let them know at the FDA. Okay, they work for us. That comes out of our tax money. Okay, and I'm sure they just love hearing that. But it's the truth. It's the truth. So okay, I'll get off my high horse. But okay, all right. So we kind of talked about that, and then that should hopefully like increase clinical trials. And how? Okay, next thing on my list I had was, does it matter as far as like what order treatments are done in?
Most, in order to make sure that you're eligible for a trial, you mean?
Well, just in general. I mean, in general, does it matter if I have surgery first, immunotherapy first, or chemo first? Does it matter? And how would I know whether I'm newly diagnosed or this is recurrent?
Well, I guess the brass tacks answer to that question is it does depend on your stage, because stage determines treatment. And if at diagnosis the cancer has figured out how to spread, it's not just in one place, then surgery isn't going to get it all. And so even if you have an aggressive surgeon that is willing to cut out all the spots, the truth is that the way the cancer spread was through the bloodstream, and so it's likely to still be there. So if you have a cancer that's learned to spread, you need systemic therapy first. So if you have a localized cancer in the lung, for example, nowhere else, then a surgical resection is typically the best way to cure it. But these days, we are also talking about how sometimes immune therapy and chemotherapy added before you have the surgery may improve your chances of cure, may improve your chances of not having to deal with the cancer again, I guess I would say.
Okay. And so
we lost our Hollywood squares.
Yeah,
I like that.
I lost one. Yeah, I lost one section. Okay, now it's back up.
There we go.
Yeah, I don't know what that was all about. Hopefully it's not. I had 1 week where it was just a total disaster every time I touched anything that technology involved. It was a nightmare. Now I completely forgot what I was going to say. All right.
Okay. Do you mean though? Terry, I do think sequencing of systemic therapies is yet another question we could talk about.
Okay. Yeah, please do.
Because I think we're in a place where we're beginning to realize that all cancers are not created equal, and there will be some who should get chemotherapy, and there are some that should get chemotherapy and immune therapy. And then there are some that should get targeted therapy. And maybe with KRAS mutant cancers, there are some that should get a targeted therapy and immune therapy. So we've known for as long as I've been an oncologist that when you see a group of people like this, you see a group of tumors that are just as different one to the other. It's a very individualized thing. And so the fact that we would think that all patients should start with chemotherapy and then move on to other things, I think is just where we are right now, but not where we'll end up.
So okay, yeah, I know that there's like standard of cares that have been established, and that's kind of like what the standard protocol would be. How do I know if I'm getting what's best? I mean, yeah, I mean, it's going to be like, if I'm just really whatever against something, how do I get that sense of peace that I'm making the right decisions along with my healthcare team?
Well, I mean, I think probably everybody on the call might have a different answer for that, but I suppose for my patients, I believe it involves shared decision making. It involves, for some, it will require and necessitate knowing that you didn't leave anything on the track. You know, you did everything that you possibly could. And for others, it is about time and quality time. There are things that people want to do, and there are people that patients want to be with. And so sometimes a very rigorous clinical trial that might be the most promising thing that a patient has access to might not fit with those goals. So making sure that your doctor knows your goals, I think, is probably the right way to feel like you're getting the best care, because then they can guide you to the appropriate therapies. I try to treat all my patients on clinical trials because I believe that that's how we are making better treatment options all the time. But I do think it's important that when patients learn about a trial that I think is good for them, that I also present a standard of care option that is also just as appropriate. Because if my mom needed cancer care, she would not choose the trial. If my dad needed cancer care, he would absolutely choose a trial. And so everybody's different that way. So I believe like a choice helps one figure out what one in one's gut is the best choice for them. And I think that would probably help empower me to feel like I had made the right decision. What do you think of that?
Well, no, I agree, but we need to hear it from you, because a lot of times we're like trying to feel through what the doctor wants us to do. Okay. As opposed to, it's like, no, it's okay that like time is most important to me, or, you know, energy, or whatever my criteria is, or maybe getting through to the next whatever. Okay. Or I have some crazy idea about something that I'm just whatever, right? I mean, everybody has their own goals, but it's okay to hear that from you and you to give us the power. And so that kind of leads us into like second opinions, which, you know, I was scared to ask for a second opinion. I'll tell you flat out, I was scared to ask for a second opinion. And my husband, who doesn't come from a medical background, was like, "Ooh, I don't know if you want to do that. You make the doctor mad." I mean, can you just kind of like sound off on it a little bit?
You want me to sound off or the panel?
Yeah.
I always, so when I was at Northwestern, and U of C had more trials than Northwestern, I told my patients, you absolutely should get a second opinion. A second opinion will do one of two things. It will either open an option for you that I didn't foresee that you had, and that's awesome, or it gives you more confidence in what I've said, and that's also good. I feel that way now. I am often the second opinion now, even for my Tennessee oncology partners, actually, and sometimes for the people over at Vanderbilt, which I always get a kick out of, that I'm at Tennessee oncology, their second opinion. But I think it only, maybe my goal now as someone providing a second opinion is to provide education, to help a patient feel comfortable and confident with their choice, and only if they're really and if they're looking for alternatives to provide those. And so I usually start my visits with, what are you hoping to accomplish from your visit, whether it's a first opinion or a second opinion. That helps frame things.
Totally. And and in the end, it was what I needed, and it didn't change anything. Okay, but it changed everything. Mm-hmm. It didn't actually change the treatment. In fact, I was NED. It didn't change the treatment, but that's where I found out that I had KRAS, and I had no idea. So, okay. Third and fourth opinions, yes, more information is better. I'm just kind of like going through the chat here, so just bear with me, make sure there's oops. Okay. Is sotorasib combinable with immunotherapy, or is it toxicity limiting it?
Yeah, that's a hot question right now. So there was data just at the very end of ASCO, a press release on Monday night, showing that adagrasib is combinable with immunotherapy without an increase in toxicity. It's a small data set as of yet, and may have something to do with that half-life piece that I spoke about at the beginning, because why would one drug that's so similar to the other be combinable when the rumors we've heard about sotorasib are that it's not as combinable with immunotherapy, although they haven't put any data in the public domain for me to know whether that's true or not.
I think that's the hard part, is the cancer for all of us is here now. Okay, but we have to keep looking to tomorrow, and we have a long ways to go to get there. Let's see, Eric, right now, let's see, Pembro, okay, timing, it's all right, okay. All right, was there anything else? Now, how about, I saw something, but I didn't actually get back to it, on pancreatic and colorectal cancers. Do you know anything around those when it comes to the KRAS? I wanted to watch, there was something about pancreatic cancer, does it make a difference? And I didn't get to it. I don't remember what happened. I was probably someplace else.
We participated here, adagrasib, in part of that same study. They had a group of non-lung patients, and we contributed. There were pancreatic cancer patients from our site and ovarian cancer patients from our site, and then colorectal cancer patients. And I would say that across the board, everybody's cancer is different, and I think we know that colorectal just doesn't, it's not enough to just give a direct KRAS G12C inhibitor. It seems like the downstream effectors turn on again very, very soon. Pancreatic is more sensitive. There was actually an early report of sotorasib data that was at ASCO plenary March, February, and the response rates were around 15-20%. So better than colorectal, not quite as good as lung. And I think ovarian, their response rates are also really good. It's a slow growing ovarian cancer that tends to have a KRAS mutation, so very different, right, from lung. And so they tend to do pretty well with the drug. So you can't apply one to the other, which in some ways makes sense to me, just because the cancers are so different. But on the other hand, if we look at a drug like pembrolizumab that Ben is on in the chat, like the fact that that works in all patients who have, a high TMB score, irrespective of the tumor type, also is pretty great, because you don't have to work so hard to find good drugs. But we'll get there with KRAS drugs too.
And then what about acquired resistance to these G12C inhibitors?
Seems like it develops within 6 months to a year. Just like for EGFR or ALK-driven lung cancers, they're programmed to grow. And so you might successfully inhibit them for a while, but then they develop, the cancer cells figure out how to grow again. And I think there are data sets actually of patients that got adagrasib or sotorasib, and the number of different types of mutations that develop, there are quite a few. So it's really important that your doc keeps looking, keeps testing the tumor. I tend to do tumor testing early and then blood-based testing at resistance. The cost of tests like Guardant360, for example, they won't run the test if your insurance is going to charge you, the patient, more than $100, and they'll call you before they run it. And so to me, that is a safeguard for ordering that test and not worrying too much about the price. Because again, if your out of pocket is going to be more than $100, they'll call you. And the other advantage is once you've ordered one test and your insurance has paid for one test within a year, then they'll comp all the other tests that Guardant360 does for you, for your profile within that year. And so I just think we continue to, as the cancer continues to learn how to grow again, the onus is on us to continue to look and see what other mutations could develop. And it probably is one of those examples where you might have to say, okay, so Dr. Johnson, when are we going to do another one of those tests? Because they may not be thinking in that way. My KRAS group thinks we should be doing it if I'm changing therapy.
How long do you usually monitor it? I shouldn't say how long. How often are you usually monitoring the ctDNA while somebody's on an inhibitor?
We have to be careful not to think that it would replace how you're feeling or what you find on imaging. Mm-hmm. Because you can see more clones develop before there's additional cancer spots, or before you have a change in a cancer-related symptom. And so I don't think that the data tells us to change therapy just because of a change in that plasma DNA. So with all of that said, I usually do it in a way that coincides with progression on a current therapy to help me assign a new therapy rather than more often than that.
Okay. And then kind of just rule of thumb, how often is somebody scanned? Like are they being monitored when they're on treatment, or, whether it's
some ways it depends on what they're getting. It depends on the risk benefit ratio, like how many symptoms they're having. But I usually scan on targeted therapies once every 6 weeks.
Okay. Yeah, because
Keytruda is an example where I think now doctors are, and I have a couple of patients in my practice, things have gone so well that I've spaced it out to once every 3 months, with the understanding and the trust that if anything changes in how they're feeling, they'll call me sooner than that. but I tend to hold people close until we develop that confidence together.
Mm-hmm. Okay. Yeah. And yeah, I mean, that makes sense. And just now might be a good time to ask this question. The difference between what you see on a CT scan versus a PET scan, and there would be a CT scan with contrast. Can you just kind of go through the highlights of that?
CT scan to me is the right test for surveillance imaging. Tennessee Oncology owns not just a couple PET scans, so we have those, and in general, I think I see patients that are that have surveillance imaging with PET. The problem is with a drug like Keytruda, that, for example, that causes an inflammatory response. Sometimes that is a little bit hard to interpret on a scan. And so a PET scan that is really measuring anywhere that there's more active sugar uptake, and that can be because of cancer, but that can also be because of infection, and that can also be because of inflammation. Sometimes it is a little harder to interpret. I can remember doing a PET scan too early in a patient when I was at Northwestern who had radiation, chemo and radiation. And I did a PET scan 3 months after radiation had finished, and it was bright on PET. And I assumed that meant that the patient had recurrent disease, and did a biopsy that showed abnormal cells. And so what does that mean? After you've had radiation, you got all sorts of inflammation. And so the patient endured a very stressful 3 months while we waited and rescanned. And sure enough, it was fine. The patient was fine. The patient did not have recurrent cancer. So I learned my lesson from that. CT scans, on the other hand, they're anatomically the best test. So if you know where cancer is because you've done a PET scan at diagnosis, for example, or at recurrence, then the CT scan is the best way to measure what the cancer spots are doing on treatment. And so that's why I use it for surveillance. Now in the future, maybe there'll be something that, there are all sorts of different radioactive contrast elements that are being used, like PSMA for prostate cancer. So PSMA is taken up by prostate cancer cells. So maybe one day that will be better than an FDG PET. But we don't have that yet in lung cancer. So I do CT scans.
Yeah, well, and they have a little bit of it with in colon cancer with the MSS and MSI.
Well, the CEA that some people will measure that CEA level. That's true. Mm-hmm. And PSA is the blood level in prostate cancer patients. But in a PET scan, it is a sugar molecule fused to radioactive iodine. That's the contrast. And so it goes in the body, and it goes anywhere that sugar is taken up, and then the radioactive iodine or the radioactive isotope glows. And so the PSMA PET is the same, only instead of sugar, it has PSMA. And so it goes anywhere in the body that there are prostate cancer cells that are interested in PSMA, and it glows. So that's a cool technology, but is starting to be used in clinical trials more. But we haven't. But I wouldn't go ask for one of those yet.
No, and I know that a lot of the proton therapy originated from doing prostate cancer treatments, as far as putting like the radioactive so that they could actually get down to those little balls and tweak it. And so I know that we've learned a whole lot from being able to essentially cure like prostate cancer, because it's very curable. I mean, obviously you can get anything too far behind. It's not, but okay. All right. Okay. Go ahead.
Terri, I'll answer this question in the chat from Robert about Melina not being able to get CT scans because she can't have a contrast dye. Because the CT scan without contrast, depending on what part of the body you're looking at, might not be that useful. It's not useful in the brain, and it's not useful in the abdomen. And so she has been able to use PET scans. I don't think that there's anything to be worried about there. Like, my boss says, don't let the perfect be the enemy of the good. So the truth is, we just need to make sure that, we just need to make sure that the cancer is not growing appreciably. But remember, that's not the only way that we make decisions about therapy or stopping or restarting. I mean, how you feel, for me, I can often tell, and we'll ask, how are you feeling, before we talk about the scan results, because that's really important. If you're feeling well and there's a little bit of growing, well, maybe we should keep going a little bit longer. Sometimes we find that, treatments are working better than we thought they were, and stopping them too soon is just about the one thing that keeps me up at night.
Yeah,
on a trial.
And I think part of the hard part, and I can't speak for everybody, but I can speak on behalf of myself, is I felt like my body misled me into thinking I was fine. And then I get diagnosed with lung cancer and I never felt anything. I just thought I would feel something and know something was there. And so I feel like I can't trust those feelings anymore. And I think that's part of it.
Yeah, that's fair enough.
Yeah. Okay. And how often would you say, since we're talking about scans and stuff, brain scans, I mean, should they be annual? Assuming like you haven't had any brain mets, or even if you have treated brain mets, I'm sure that there's going to be a distinction.
If you haven't had brain mets, then I would say at progression is a really good time to look for brain mets, whether you've had them before or not. If you've had any growth in the body from the neck down, then I want to look up above the neck as well. If you have a cancer that has figured out how to be in your brain, somewhere between every 3 and every 6 months is when I scan. For an ALK positive patient where, the brain tends to be a sanctuary site, I'll scan it. I'll scan every 3. So I'll do a CT scan at 6 and then a CT scan and a brain scan at 3 months.
Is there any tendency in KRAS cancers one way or the other with brain?
Because KRAS mutant cancers are so variable, I wouldn't say that your predilection is any more or less. 30% of lung cancer and 90+% of pancreatic cancer. That it's another place where I use how patients are feeling also as another reason to look. And then finally, again, I'll use the clinical trial excuse, but most clinical trials will require a brain scan. Patients that come to me from, outside Tennessee oncology are typically like, yes, please scan my brain, just to make sure. And I hate that they feel that way because, there doesn't need to be that reticence to scan before clinical trial participation.
Yeah.
There are parts of the country where getting a brain scan approved is more difficult than others, but we're fortunate in Tennessee.
And it does matter. It does matter. And sometimes not for no reason other than it just for some reason matters from one place to the next with insurance. Okay, you see brain scan and not brain MRI.
No, brain MRI is the way to go. I don't want you to take away that Dr. Johnson doesn't do brain scans, but for reasons that, you've articulated, sometimes you can't have a brain scan, or you can't have a brain MRI, pacemaker, ICD, that kind of thing. And then a CT scan with contrast is okay, but a brain scan is much better. Brain MRI is much better.
Okay, I'm scrolling through this. What I'm doing is looking through the chat, see if I missed anything over here. If anybody has anything, like, go ahead and dump it into the chat or raise your hand or something. Okay, well, I mean, so that's
a tough one. Here with the utility of liquid biopsy vis-a-vis PET and CT scan.
That's a good question.
I mean, you know, you get very quickly into these medical terms like sensitivity and specificity, which is if you see something, is it definitely cancer? Like the PET scan, if you see something, it might not be cancer. Sensitivity is very high versus specificity being high, where if you don't see anything, the chances that the test is positive is very low. Or you know, just because you don't see something doesn't mean that you can bank on it. Like, and these parameters exist for all these different tests. So liquid biopsy is good if it's positive, but if it's negative, it doesn't mean that you don't have cancer. CT scan and PET are, in particular, are better for looking to see if there's any cancer in your body. Sometimes CT scan better than PET. Well, once you've diagnosed cancer and you know that it's there, PET is good for diagnosing, in particular in the lung, like occult little bits of cancer in the middle of the chest or cancer outside the chest that you might otherwise have missed. So they work together, I would say. I think of them kind of like a Venn diagram, right? You get complementary or supporting information from each one. But sometimes one individual test doesn't give the whole answer.
And I think there's a lot of that kind of just goes across. All right, Lori's got a good question. So if someone has had a complete response on one of these drugs, what is left to resist? What is left to resist? In other words, she's had complete response on an inhibitor. Blah, boy, on an inhibitor. Okay, so on a G12C inhibitor. Okay, what could go wrong or what could go right? I mean, is it possible that that's it? She's just, there she is. She's done. She's NED and she's just going to move on with her life.
Well, geez, that's a tough question for 4:30 on a Monday afternoon. You know, I tell my patients my crystal ball's in the shop. You know, I get, you know, Lori, I don't know. I have to, unfortunately, as a medical oncologist, we're taught that when a cancer learns to spread, it's hard to get rid of it completely. But these drugs are kind of a new frontier. And depending on the sequence, depending on what you had before you went on this drug, depending on what your doctor has done subsequently, like radiation, I would say that there's hope for long-term, durable, disease-free living. And, you know, I'm always happy to be wrong about being worried about cancer coming back. But, you know, I don't get paid to always have the happy story. You know, I get paid to think about the best case scenario, the worst case scenario, and present you with all of it so that you are informed. And then the hardest part is the living in between the variables that you don't know where the bounds are.
That's pretty much how life is. I think we just don't know that there's boundaries until we get a diagnosis a lot of times. I think that's a lot of it. Okay, yes. Mandy's the question. If you sign up for a trial, but you don't get selected for the trial drug, just the standard of care, your cancer is progressing, is there an appeal process to get on the trial drug? Or?
Well, remember that, you know, a lot of these trial drugs, the reason the FDA requires a randomized test for I would say for some of them. And I think it has to do, I've never worked at the FDA, so I could be wrong about this, but I think it has to do with the response rate. For example, osimertinib's response rate is 78 to 80+% for patients in the front line. And so that's better than chemotherapy alone. So the FDA didn't require randomized test to approve the drug. They did require one to make the accelerated approval become complete. So we would hate, and this has happened, by the way, we would hate to assume that a targeted drug is better than the standard of care and it not be. And so that's the reason for the clinical trial where you are randomized to receive, the half are randomized to receive, not placebo, but typically these days, a chemotherapy. I love the trial options that let you cross over, and I ask for them all the time. And honestly, a reason to go on a phase 1 trial these days is because that's where a lot of the work is done. I do have many patients that come to me and say, oh, I don't want a phase 1 trial. And I say, oh, but you do, because we know so much more about you now based on molecular profiling, based on what you had as you got chemo and immune therapy, presumably already. Like those are the only standard of care options that bring longevity with good quality of life. So now we can match you to a trial that makes sense for you. And you can get it in a phase 1 trial as the only drug. Whereas then there is a randomized phase 3 effort, typically against docetaxel, you know, and that is a really bitter pill, I think, to swallow sometimes when you're really motivated by the clinical trial drug. But on the other hand, in my lifetime, there have been drugs that we thought were better than docetaxel that weren't. And so I wouldn't want my mom to be on a drug that didn't have an approval based on data. So it is very tricky.
There's no easy answers, are there?
No easy answer.
Well, in the phase 1, yeah, Alyssa, I thought phase 1 trials were for toxicity as well. And so
not anymore. It is a part of what they do now. But because clinical research is so expensive, the drug companies want much, much more information from a phase 1 trial. They want to understand the half-life. They want to understand the side effect profile. Yes, but they also want to know if it works. And so typically a targeted therapy for a group of patients that have a particular mutation that's driving their cancer, we say, they are absolutely looking to get a response rate from that group. And if you think about it, if you enroll everybody to an unselected trial or to a targeted therapy drug, like I wouldn't feel good about putting a patient without a KRAS mutation on a KRAS inhibitor anymore. And in the past, that's what happened in phase 1. But that doesn't happen now.
Yeah. And I don't know. I think I'm still kind of, ASCO was my first in-person ASCO. So this was the first time I was there in person, and I had a lot of fun. It was a little bit more tiring than I expected, but, and I'm still kind of like, you know, drowning from all the information that just kind of like came out from it. Did you see anything else that was just like, it's really compelling, you just need to know about this or hear about this? And it may just be like a cancer thing or whatever. I can tell you that, okay, so along these lines, and if somebody has a question, just hit it up in the chat too. But, you know, as slow as things feel in the States to get FDA approval, when I see the comparisons to other places in the world, I'm always surprised by how much faster it happens in the States than it does other places. And one of the presentations I was at, they were talking about the treatments that they get in India. And just, it was something insane, like less than 3% of the population who qualified for an immunotherapy get immunotherapy because of the way that you're paying for it out of pocket. And I don't know, a number of different reasons, and awareness and diagnosis, and a lot of reasons. It wasn't just simple. And it was like, you don't even realize how amazing it is that we even have this kind of like legitimate hope seeing out there, until you look at something else and you're like, wow, I mean, wow, that's crazy.
Mm-hmm. I agree. We are lucky in the United States because of the amount of money that we spend on drugs. That is money that then goes into research and development at those companies. And so they can do it again. I think that there probably needs to be some restriction. But I think in the United States, we have also been reluctant to impose any restriction on any drug that would potentially help a person, because there's a lot of liberty in that. And so I think politicians and physicians are united in not wanting to ever say no to that prospect for a patient. Clinical trials are a little different. So let's see, any big picture takeaways. I guess I took away how important one-on-one collaboration is, or like face-to-face collaboration. I mean, this is good. This is better than than nothing, certainly. But I realized that so many of my partners that in pharmaceutical companies, also in the CROs that sort of manage the trial conduct, we fight so hard for our patients. We fight so hard to get everybody a fair shake. And, sometimes I feel like on the other end of the phone call or on the other end of the Zoom, they are just clueless to what my life is like, taking care of people with cancer. And so it was good to be in a place where we could connect a little bit more. So you realize you are everybody is on the same page and trying to help patients. And everybody comes at this problem that is trying to cure cancer from different perspectives.
Yeah, I think we all are. And I think, the general theme I kept hearing is something we've been hearing since, the last couple of years, about diversity. And I think that there's so much more to it than, boy, I can't even just pull it together today. There is just like so much more to it. Okay. As far as like you were saying, you can look across the room and see totally different people. And even though we may even all hit the exact same mutation, it's going to look differently within our body. And we're going to have different metastasis, different co-mutations, and different color eyes, and different color skin, or whatever. All these different sorts of things that go on, and blood types, and all the other things that are different, even though we share this one commonality. And consistently, I kept seeing that we need to get more diversity across the board, as far as every different walk of life, like every different walk of life, whether it's, location or finances or race, creed, color, I don't even know, whatever, all the different parts of of life that are involved. And there's going to be more to it than that. So, I mean, that was really we've been hearing it a lot, but I think hearing something when you're sitting in a Zoom room with 200 people or 20,000 people is completely different than being in a room full of 10,000 people that kind of have the same sort of a takeaway.
Yeah.
So, yeah. Is the Astralis G12D drug oral, is the question that just got hit up there. I don't know.
I believe so. I believe so.
Okay, and all right, well, it's like 5 minutes till the hour, and you're very, very generous with your time. So I'm gonna like throw a couple last minute questions out there, and see, make sure I'm trying to scan through the chat, make sure I didn't miss anything. CT with contrast versus MRI pros and cons. I think we covered that, right? Didn't we do that? Didn't we talk about that? Yeah, yeah. Okay. All right. Do you have are you like recovering still from ASCO? You still have your like science geek on? How does this work?
Well, you know, that I guess the dirty secret, Terry, is that life goes on here in the clinic when we are at ASCO, and the nurses and the nurse practitioners especially love to remind us of that this week. And so I have to say that I am always rejuvenated from ASCO and come back and make lists of things that I want to do better, things I want to do differently, things I want to share with my team in the clinic. So now's the hard part to, you know, to tell everybody about it that didn't get to go. We did that today. We had, we recognized every team at Sarah Cannon whose study became an oral abstract at the meeting. So there were 6 orals just from Sarah Cannon in Nashville. And so we got, so we recognized every lab person that touched the patients who were on that trial, and every tissue person who had to do something with the tissue for patients on the trial. And then we did this big lottery and like pulled out names and had gifts. And I did use some of your KRAS socks for that. It's really important. I realize in this year of COVID and the Great Resignation, where so many of the people that work at Sarah Cannon are young people, that we have to be real diligent about helping them understand how impactful their work is. Like, it's easy for me at a meeting like that, but it's really important for the people who work here to understand that, because that's what makes them keep coming to work, or, you know, working from home post-COVID, but understanding and believing in the mission that we have here. That's what makes people stay. So that's what I'm worried about now. That's what I'm preoccupied with.
Yeah, I can see that. See, when I was at ASCO, I felt guilty because I couldn't be talking to my patient community, because I felt like I was there trying to absorb all the other information and make the connections that I needed so that I can get people to come and do the crossover. And so again, it's like, and as a patient, there's a part of me that just feels horribly guilty about, I can't be everywhere at once, right? I just can't be everywhere at once.
You're right. And I think you can't listen to you can't really pay attention to more than one thing at a time. I've had to learn that lesson. Really, truly, genuinely pay attention in a way that, you know, you will, there are some people in the world, I work with a couple of them, but I am not that way. So I don't think you should feel guilty, though. I think you should feel like you are the representative. And maybe next year there can be more representation.
I hope there can be more next year, because it was really fun. And AACR was an absolute blast. I had so much fun at AACR. Now, I'm not sure if that was because it was the first time I stepped out after COVID, and it was really great, or what. So let's see, I think something, has there been any COVIDs? See, speaking of COVID, Eric, you've been busy with lots of questions today. Speaking of COVID, has there been any studies of how any of these targeted therapies, drugs interacted with COVID vaccines versus unvaccinated?
That's a good question. This was the first meeting where the number of COVID infections was sort of part of the statistics that were reported. Not in all the studies, but in some. And in general, Terry, I don't know whether you were struck by this, but I guess I was struck by the fact that there weren't more. And I think it may have to do with the fact that where the patients were being enrolled were not places where the COVID infection rate was real high at the beginning. We continued, at least in clinical trials, right? Patients still needed to be treated, but we were fortunate that we just didn't ever stop enrolling patients to trials. We didn't stop coming into work because Nashville didn't have the numbers that New York City did, that Boston did, where they had to sort of turn down the faucet for a while. There are some studies that suggest that patients whose immune systems were compromised because of chemotherapy maybe had a harder time with COVID infections, but it didn't seem to be the case for immunotherapy. And it didn't seem to be the case for targeted therapy that I'm aware of. And I guess that makes sense.
Yeah, well, I don't know. It constantly surprises me how when I see all this data and all these brilliant people putting up these slides, you are either a never smoker or a smoker of smoking history, not necessarily a former smoker. And so there really needs to be 3 categories regardless, because somebody who ever smoked a cigarette does not really necessarily be somebody who's currently smoking. And I haven't even seen that consistently applied. So the COVID thing, I don't know where that's going to go, and that's one of the, that's a real burr under my saddle, is that doesn't get split out, because it makes a world of difference. I'm growing up when I did in the 70s and 80s, the entire world smoked. So either people are lying or they've smoked at least the 10 cigarettes, okay? Because that's the world that we were in. And so it's just, it's not accurate information otherwise. But what orals did the other folks from Sarah Cannon present? What were some of the...
There was one oral in our section, in our, or in the abstracts, the developmental therapeutics that was after ours. And it was an antibody, it was Erica Hamilton presented an antibody drug conjugate directed against CDH6 for kidney cancer patients and ovarian cancer patients with great results, responding patients in terms of shrinking tumor lesions as well as CA125. I presented an oral abstract on a novel immunocytokine made by Takeda called Modakafusp alpha that has been studied in myeloma patients and now being studied in solid tumor patients, probably more in melanoma. We had, an interesting targeted therapy against a mutation TP53Y220C. That was part of that developmental therapeutics section as well. I have a small cell patient on that trial who has was the best responder in the trial, has done really well. And that was a surprise. Like, we thought that was going to be such a needle in a haystack group of patients to find. But when you look, you find
any response in small cell is miraculous.
Yeah.
It's kind of like any progress in pancreatic cancer. I mean, it's a big deal.
And really probably underscores in the last minute of our time together, just the awareness about targeted therapies, the amount of research going into identifying patients and developing drugs when, mutations underlie your cancer. So there's a lot to be hopeful for.
Oh, there is indeed. We've on our t-shirt says hope shared is hope multiplied, because it's the truth. You share it, it multiplies it. And so we're just going to keep on growing it. So I'm going to use that to kind of like wrap things up. But thank you so much for being here today and answering the questions and being, just direct and approachable and, straight up with us. And so I thank you so much for being here. And I'm sure we'll be seeing each other again soon in the near future. I guess I'm just going to close us all out. So
thanks for the opportunity, Terry. And thanks everybody for staying on to the end. And thank you for my socks.
Oh,
they brought a lot of people joy. I can't even tell you, I can't even tell you who's been the most excited. So thanks for the socks.
Oh, for sure, for sure. Okay, cool. We'll just keep on rocking, keep on kicking cancer's KRAS, right?
Okay.
Okay, cool. All right, bye, y'all.